Hello everybody, hope all of you are doing well. So today in this video I'm going to discuss with you a very simple formula to remember the structures of the amino acids. In a basic layman language I can say that the building blocks of the proteins are called or are known as the amino acids. They form peptide bond and they form a long sequence of amino acid actually to form the protein. There are different stages of protein formation. So I will not go into a detail, but this video is for those who actually want to recall and remember the whole structure, all the structure of all the kinds of amino acids that are actually exist. Basically, I will talk about a very few of them, although, but in this video, I am going to discuss a very simple formula, a very simple trick to remember the structure of the amino acids. So, without any further delay, let's dive into the topic. So, as you all know, the amino acids in a very neutral pH, they actually have no charge. So, they form the zwitter ionic structure as I have shown over here. So, the there is actually the COOH and there is NH2, but in a pH neutral, they actually forms this kind of ionic structure, which is actually also known as the zwitter ion. I'll not go into that detail of this, but with this help of this structure, I will show you how to remember the structure of all the kinds of amino acids. So here is the R. As you can see, I have written a R. So I'll substitute this R with different groups to show you how we can remember and how we can form different amino acids. First of all, I'll come over here. As you can see, I have substituted the group R with H. This is actually known as glycine. The simplest amino acids exist in the planet Earth. Moving to the next, I replaced this hydrogen with the methyl group. Okay, this is alanine. Okay, for very easy format, if you can remember your class 12th basics, we have remembered this formula, right? Formic acid, acetic acid. Remember, I guess. So you can think sort of like this thing over here. Moving to the next, if I'll show you this alanine, from this alanine, I'll just add CH2. Okay, I'll just remove one hydrogen from them and I will add a phenyl group. This is known as phenylalanine. Okay, from this phenylalanine, if I'll draw again the phenylalanine and just I will add a OH over here, just a hydroxyl element radical. This will transform it, sorry, tyrosine. You can see we just got 4 different amino acids. Now I'll move to the next page. Now let us again come with the alanine. Okay, this is alanine. Now what I will do, I will again draw the alanine. Sorry, I'll again draw the alanine. I'll replace one H with the SH. Okay, this is known as cysteine. Okay, now the fun part is that you see here is a hydrogen over here. Okay, if I'll remove this and I will replace with this, if let's say this is a CH2 S, and then if I'll again do the same thing sorry. If I'll do like this, and I'll make the CH2, you can see they are actually forming a dimer of cysteine acid, right? This is called cysteine. Okay, this is actually dimer basically. And if I'm wrong, sorry. Yes, they are actually forming a dimer of the cysteine. Okay, and again, if I'll take this cysteine and I'll replace this hydrogen with a methyl compound SCH3. If I'll replace this hydrogen with a methyl, I will get methionine. Okay, so remember, again I am saying, from alanine, if I'll replace one hydrogen and I replace with SH, I'll go cysteine. Okay, and then if I'll make a dimer of cysteine by replacing this hydrogen, I'll get cysteine. Okay, and from the cysteine, if I'll replace this hydrogen with CH3, I'll get methionine. Methionine, if you know, the AUG codon, which is the starting codon of the genetic code, that actually codes for the methionine, and this is the starting, as it is the starting codon. So if you in a very general idea, I'll say every protein contains sulfur. If it is methionine, it is the starting of every protein, so hence every protein contains sulfur. Okay, moving to the next. Again I will draw alanine. Okay, I'll go to the next page. It will be very good for me. This is alanine. Now I will just do this thing. What I'll do, I will just replace this hydrogen with a acidamide, CO CH3. Sorry, sorry, CONH2. Then what will happen? I will got aspartame. Just see this thing, and if this I'll just paste another one. Now from this aspartame, I will just replace this thing with COOH, which is actually aspartic acid. From this, if I'll add a CH2 in between them, let's say I'll add a CH2 between them, so in this time I will be writing CH2 COONH2, that is glutamate. And from this glutamate, if I'll remove this element with COOH, if I'll replace that CH2 COOH, that is glutamic acid. Yeah, it's sort of very difficult sometimes to remember these whole things, but you need some practice first of all. And second most important thing is that you need some basics of organic chemistry with this thing. Okay, you need to know what is acidamide group, what is this thiol group. Okay, SH is actually known as a thiol group. Okay, So you need to know these things, very basic things in your organic chemistry. These are basically class 11 and class 12 standard, sorry, class 12 standard chemistry. So if you know these things, you can remember each and every amino acids like this. Okay, and I am just making them from this whole alanine. I'm not going in any other amino acids. I'm just making them from this alanine. Okay, moving to the next. Let me draw one another alanine, and then what I will do, I will just replace this CH2. I'll just make CH2 CH2OH. It seems like I added a methyl alcohol, right? It's this formula methyl alcohol is CH3OH. I'll just add it, just I remove the H and I just added to the chain. So this is actually known as serine. Just let me do one thing. If I'll do this again, same thing, again same thing, and I'll replace this OH with COOH. Just let me draw. Then it is threonine. Okay, so you got alcohol amino acid. Okay, the alcohol containing amino acid. Again, this is a tricky one, which is a CH3 alanine. For alanine, I'll replace this thing H with this ring. Okay, C single bond. This is a tricky one because this amino acid you can't remember with any formulas. I didn't find any formulas on this thing, but if you can remember like this is actually tryptophan. This amino acid is having a different formula. I tried to search of any idea for remembering this one, but I didn't got anything. So this is the tryptophan. For alanine, I will just replace this CH2H with this with ethyl amine. Okay, so CH2 CH2 NH2. As you all know, the formula of ethyl amine is this is the primary amine. Okay, although this is the primary ethyl amine. So if I'll replace this is called lysine. This is also an essential amino acid. Okay, and from this, if I will replace the same thing with a tertiary amine. Okay, so here what will happen, I will replace this thing with a tertiary amine, sorry, NH, C double bond, NH, single bond, NH2, then it is actually arginine. Okay, so these few amino acids that I tried to make a very good idea to remember. Although this is my own notes, I remember these things like this, and this actually, the first thing is that to remember or understand this whole theorem or whole thing, you need to know class 11 standard and 12 standard basic chemistry, because you need to know the groups. Okay, if you do not know what is methyl group, you do not know what is ammonia, what is amine, you couldn't even recall the formula. So for remembering the formula, the only art is that you need to master the groups, and that is how we can remember the structure of the amino acids. So again, this is all about my video. Thanks for watching, guys. I hope you like this video. Thanks a lot.